So, good afternoon. Well, hi. Hello, everybody. As far as I can see, already high school students are here. So, first of all, I would like to thank the organizers for inviting me for this speak. In this talk, I will present our research group's projects, which you can see here. So we have been currently running various projects on viruses and humans, and we are about to start working on the microbiomes. As for the viruses, we are examining the dynamic transcriptome atlases of the viruses using cutting-edge technologies, and we also use these organisms as models for the investigation of the general aspects of the genetic regulations. And also, we use them as tools for mapping of brain circuitries. So due to the limitation of time, I will only focus on the viral projects and try to explain it very briefly what we are doing. So first, let me start with the brain circuitry tracing. So for that purpose, we use a herpes virus called pseudorabies virus, PRV in short. And this virus is able to cross the synapses within the central nervous system and also the peripheral nervous system. And in contrast to, do we have a laser pointer, by the way? Because I would like to. Okay, thank you very much. Okay. Right. Okay, so I have to, okay. Right, so in contrast to the traditional tracers, which are dyes, the herpes viruses are able to cross multiple synapses. So we can trace multiple synaptic pathways. So the point here is that if we can trace the viruses within the central nervous system, then we can correlate it with the functional connections within the brain. But there are several problems with the white-up viruses. We have to genetically modify that in order to obtain an ideal tracer vector. For example, one of the problems is that the viruses are able to spread both in the retrograde and also at the anterograde manner. This is a problem because it would mess, the specificity of the labeling. The other problem is that the white-up virus degrades the cells, and as a result, it will get or released into the extracellular space. It's also a problem because the specificity is in stake. So we genetically modify the virus in order to eliminate these inadvertent properties. I don't go into details how do we do that. So, right. So we have developed several valuable viruses for the brain research. So one of them is called as the rainbow viruses. The rainbow viruses are individually equipped or integrated different type of fluorescence proteins. So if we inoculate different parts of the brain, then we can trace them. For example, if you see at this nuclei, which is called the locus ceruleus, so if we inoculate various brain regions, then the messages here will be integrated, the viruses will be integrated, and we will obtain a fine structure. So we can ascertain here how the different parts of this nucleus, controls the other nucleus in the brain. Another type of viruses is the so-called timer viruses. In these viruses, we integrated two different color-expressing fluorescence proteins, the green and the red, and we encoded these proteins in such a way that the green appears 6 hours earlier than the red one. So why it is good? This is good for two different reasons. One of them is that using these techniques, we can ascertain the synaptic connectivity, the synaptic orders of the neurons. Sometimes we don't know which neurons in a certain brain center, the sending neurons, and which one is the receiving neurons. But if we trace them, you see here that this is a late infection, and this is an early infection. Late because the red fluorescence proteins has already appeared. So the progress of the virus is that direction, and this is in contrast to the spread of the action potential, the information. So we can predict that this is the way or the route for the progression of the neural information. Another thing what we can use for this timer viruses is that we can monitor the health state of the infected neurons. So if an infected neuron expresses exclusively the green color, we can say that this is a healthy neurons. When the red appears, then we cannot use the cells for functional analysis. Maybe the most interesting viruses are the so-called activity sensor viruses. So that viruses express so-called fluorescence activity sensors. Here you can see this molecule. So that this is composed of two fluorescence protein genes. One of them is blue, cyan, the other one is yellow, and this is linked with a calcium ion sensor peptide. So we know that the high calcium level in the cytoplasm is an indicator of the cellular activity. So when the cells are active, then these two subunits, these two color subdomains of this protein get into the close proximity. And if we induce the blue, then there will be also yellow expression. And the ratio between the blue and the red can be calculated by a computer, and tell whether the cells are active, and if so, what is the level of the activity. The beauty of this system is that we can monitor a large number of neurons. The activity of neurons can be monitored using optical methods. So the other topics which we are dealing with is the assembly of the dynamic viral transcriptome. So in this study, we use the cutting-edge technologies such as the third-generation sequencing, Illumina sequencing, etc., etc. So as you can see, we examined really a wide range of viral families with a lot of members. And what we obtained was very interesting. It turned out that, so actually we were the first to use the third-generation sequencing. What is the point for that? The third-generation sequencing is able to read the entire transcript, entire RNA molecules. This is in contrast with the so-called second-generation sequencing. I will show you a picture maybe much better. So it turned out that with this technique, this third-generation sequencing techniques, we could multiply the number of the previously known RNAs. So in many cases, we obtained 10 times more different transcripts than it was known. So I can say without modesty that we could rewrite the viral transcriptomes, actually. So for the illustration, I can show you a very simplified version of the herpes simplex virus type 1 transcriptome. Here are the genes. As you can see, a single gene codes for multiple transcripts, or a lot of transcripts. Actually, I said that this is very simplified because there are much more transcripts. So what kind of transcripts we obtained? So first of all, there are genes, right? So the genes contains the coding sequence starting with the ATG or AUG on the RNA, and there's the stop codons, and this is the coding part of them. It turned out that practically every gene has an embedded smaller gene producing a smaller protein molecules. So why this information had not been detected before? This is because the old-fashioned technology was the short-read sequencing. As you can see, I mean, from this pattern, you cannot say that here is a gene and here is another gene. You can see it. This is in contrast with the long-read sequences. You obviously will see that there are two different populations of the transcripts. So these are the coding sequences. We also discovered a large number of the so-called non-coding transcripts, and you can see what kind of variations. So there are 3' truncated transcript, intergenic transcript, and trisomic transcript, a lot of different transcripts. And there are also various so-called transcript isoforms. So it means that we talk about the same transcripts, but their termini are different. They are longer at the 5' end, and this is called as a transcriptional start site, or they are longer at the transcriptional end site. And there's a wide variation of the splicing patterns. So using the short-read sequencing is actually blind to this variation. Long-read sequencing is needed to detect, to identify this kind of isoforms. Here we can see a concrete example. So these are the sequencing grid. I don't go into detail. What you can see here is that this is the normal, transcript of the genes, and this is a longer one. So this is a transcription start site isoform. The transcription end site isoform, I'll show you two examples. One of them is a baculovirus. The other one is a herpes virus. As you can see here, that this gene is much longer than this one. Normally, the complexity is much, much higher. I only show you two examples, but normally there are much more transcripts of the same genetic region. This is the same for the splice isoforms. There's a smaller one and the larger one. Actually, in a normal situation, there are 10 or more spliced isoforms, and earlier it had gone undetected because of the shortage of the, or limitation of the old-fashioned technologies. So especially our technology was very suitable for the identification of the so-called multigenic transcripts. So there are two kinds of them. One of them is the so-called polycystonic transcripts. Polycystonic transcript means that the messenger RNA contains multiple genes. This is not typical at the eukaryotic organisms, typical for the bacteria. The viruses use this logic, or I would say use this polygenic transcripts. At this moment, we don't precisely know the reason for that, because only the most upstream genes are translated. We have an idea, I will tell you at the end, why these genes are also expressed. And this is another type of multigenic transcript. We termed it as complex transcript. We introduced this term into the virology, I would say. Complex transcripts are composed of multiple genes, but at least one of them stands in the opposite direction to each other. So it means that at least one of the genes is meaningless, that it doesn't encode anything. Here we can see, for example, a polycystonic transcript. This is a long one. There are two short ones. Complex transcripts can be found in a pox virus. This is the vaccinia virus. As you can see, this long transcript encode multigenic transcript. We also can monitor the dynamics of the viral infection. So if we take virus samples from different time points, then we can examine how the different isoforms are produced. Here I only show you the transcription start site and also the transcription end site. So it was known earlier, and actually we published it earlier with old-fashioned technology, and this is the new techniques. As you can see, the whole transcriptomes of the viruses have been completely rewritten. And here I show you just very few examples. For example, the changing of the splicing patterns during throughout the time of the viral life cycle, changing of the 5' UTR, 3' UTR, etc., etc. There's a certain dynamics, and we are working on to figure out what could be the functional significance of the utilization of different isoforms at the different time points of the viral infection. So the major discovery what we made with this virus is the vast complexity of the transcription overlap. So this was very surprising. So actually there are no genes which would not overlap with another gene at the transcript levels. And not only the adjacent, the neighboring genes, but also the distal genes, It means that, for example, if we talk about convergent genes, that these genes, here. So in many cases, the transcription stops here, but a certain extent, a certain frequency that writes, the transcription does not stop, it continues. And so that kind of transcription overlap forms a very, very complex networks within the viral genomes. Every virus what we examine exhibit the same pattern. So why? What is the reason? The viruses, are tiny creatures and very economic creatures. Why they waste their time, for producing multigenic transcript and such kind of transcription overlaps? This is because there is, at least according to our hypothesis, a new level of genetic regulation, which we called it as transcriptional interference network. It means that the activity of the genes has an impact on the activity of the neighboring and the more distal genes. And that is why we are dealing with this large amount of the virus, is to find evidence that this is the case in order to examine the natural, the endogenous behavior of the virus, and also by modifying the virus using the genetic engineering technology. So we introduced two ways of models. One of the waterfall models. It means that one direction, so only this gene affects that gene. So this is a unidirectional interaction. Or there's a seesaw model when the two genes, both of the two genes, interact with each other. And this can be a convergent or divergent, complex overlaps, etc., etc. So if we examine, for example, the convergent genes, we can see that the dynamics of the expression of the RNAs, exhibit a complementary pattern. So we assume that when one of the partners of the convergent gene is active, that it exerts a negative effect on the other one, and vice versa. If we insert a very strong viral promoter into the viral genome using gene technology techniques, then what we obtain that expression of these antisense RNAs and also the process itself will reduce the activity of these three genes. So what happens here? It is very likely that the transcriptional machineries, collide with each other and they modulate their expressions. We discovered a new class of RNAs, non-coding RNAs, or different isoforms of the coding RNAs, and we termed that as a replication-associated RNAs. Whichever viruses we obtained, we found that there are RNAs which overlap the replication origin. The replication origin is the starting site for the replication, and it was very surprising. So, for example, in the herpes viruses, the replication origin, can change from species to species, a different position. And it doesn't matter where this replication origin is located in the genome, that overlapping always happens. So there is a function for this transcriptional overlapping of the replication origins. Here is our hypothesis. For example, the herpes viruses has two kinds of replication mechanism. They start in a so-called tetratype manner. So the replication fork progressing in two directions. And this is the initial form of the replication. And then comes the replication-associated RNAs in function. So one of the directions will be blocked by this activity of the replication-associated RNAs. So this is the two direction. When it is active, then it will block it. And on the other hand, the other replication-associated RNAs open the double-stranded DNA, thereby facilitating the progression of the replication fork in a unidirectional manner. So that is the hypothesis, and we call it the transcriptional and replication interference network. And this is only the first step to determine when the replication to start and in what direction it should be continued. There is also another aspect of this, but we are starting to run out of the time, so I have to finish this. And finally, we also found some evidence about the polymorphism of the transcript. What we found was that there is a shorter variant of a transcript and a longer one. And in the longer one, we always, every case is found the so-called upstream ORFs. The upstream ORFs, we can call that as a tiny gene or something like that. It has been shown by others that these upstream ORFs exert an effect on the translation. So we assume that this polymorphism within the transcripts play a significant role when and how the transcripts are translated, because these isoforms are expressed in a differential ratio at a different time point of the replication cycle of the virus. And I forgot to mention that the other project was acknowledged by a very prestigious journal to publish an article. It was the Nature Method, and this project is also acknowledged by the scientific community by inviting us to produce a review article in the Trends in Microbiology. So here you can see the summary. I don't want to go much into details. If you have any questions in the meantime, you can look at this list and then you will understand what we have done in our research. Thank you very much.